Wait, there's one question. Um, I think Professor Michael, there's Professor Shandar on a Sunday afternoon. Thank you so much for joining us. And to introduce Professor Shandar, he's a professor and scientist at School of Computational and Integrative Science at JNU. And he works uh, on bioinformatics, big data analytics, but his exemplary work in research in developing tools, which is on protein, DNA, protein RNA, protein-protein interactions are extremely highly cited. And apart from that, I think one of the aspects which I immensely respect of Professor Shandar being an exquisite poet, but I'm looking forward to hearing the science part today. And over to you, Professor Shandar. Thank you.
Thank you. Can you hear me?
Yes, you're audible.
Yes.
Yeah.
Thank you for keeping the introduction brief, and I was patiently waiting. And it's a great pleasure. I start with, since you introduced me the poet, which has started happening recently for last 1 or 2 years. Before that, it was kind of my private hobby. So I start with one thing about the topic that has been assigned to me. Like यूं गम छुपा रहा हूँ जैसे नया रईस अहबाब को भी अपना पुराना पता न दे. When you become newly rich, you don't want to tell your old address to anybody, even to people with whom you are spending time. And when Professor Raghav and all those who have been your buddies, who have been your mentors or your leaders in the field, and they know your old address, so they will always remind you of your old address. That is the work that we have been doing in the past. So it is kind of when we are invited to talk about molecular interactions. This is in some sense a nostalgia of what we have done in the past. Maybe our fields have digressed a little bit from there, but it's wonderful to be recalled and to understand where in that field we have moved forward over the years. So today, I think it becomes a perfect prelude that Michael, who also has been a very close friend, a senior friend who has introduced me to many of these topics in the past, and that he built a background of protein-protein interactions, and now we go to the protein-DNA interactions because the number of this makes a perfect complement. And many of the ideas that Michael has shared with you will also be applicable in this scenario, but I'm putting it, taking it a little different way. Some of the slides that I'll show you, may have already presented in Triple IT. So those of you who have already seen these slides, please forgive me for that. And I hope maybe some corner of that slide will be new to you and you can find something different from what I said last time. So first of all, protein DNA interactions are the key components of life's molecular circus. So if you think of life as a circus, and in the circus, what happens is that there is a perfect coordination between one player and the other player. And if you make a little mistake in that acts, in the kind of acts that are assigned in that circus, that can be fatal. And that can be very dangerous. So a perfect interplay between molecules, their interactions in a predefined manner is very important. And if anywhere a mistake is held, if one player does not hold the hand of the other player at the right time, this kind of mistakes can be severely detrimental. And that is the case also in the case of the molecular interactions. If a molecular interaction does not take place as it has been programmed, as it has been expected to be in the life's program, then it will lead to disasters, diseases, death, and all deformities, many different kinds of things. So there are protein-protein interactions, there are interactions with metabolites, there are interactions with carbohydrates, all kinds of molecules. But even before a protein becomes available for interaction with some other molecules, it is basically a product of protein DNA interactions, because it is the transcription factors, for example, that will interact with the genomic DNA and tell the genome to help in producing a particular protein. So, but transcription is not the only scenario in which protein DNA interaction takes place. Although when we think of protein DNA interaction, transcription comes to mind first. There is also the role of protein DNA interactions in recognizing pathogens. some of the pathogens have a kind of DNA which is not found in the hosts, in the humans, for example. So the immune system can immediately recognize if a new DNA comes, and if this DNA is a different DNA that this particular organism is familiar with, it can immediately tell that the attack has taken place and immune system has to be activated. So this is about pathogen sensing. In the innate immunity, there are specific nucleic acid sensing molecules, and there are DNA sensing molecules. Then you have in the packaging of the DNA, when the interaction takes place, basically to preserve the integrity of the genome and to also do some corrections if during the replication process, etc. And the methylation process, acetylation process, the epigenetic factors. So these are, broadly speaking, the 4 different scenarios in which protein DNA interactions take place in the eukaryotic life, to define it specifically. Now, what does bioinformatics do in the case of the protein DNA interaction? How can bioinformatics help? Of course, protein DNA interaction exactly, or for that matter, any biological interaction, biomolecular interaction, will actually be discovered only in the laboratory when you do an experiment in the cellular environment. What you do in the computational environment in silico is often not followed up or not backed up by the in vitro analysis when you do it in the test tubes. And what you do in the test tubes does not fit very well with what you observe in the cell. And even then, if you have the animal models, what you do in the mouse, we trust this will probably happen in human, but it does not. So there is a lot of cost involved in going from one low confidence information to high confidence information, and bioinformatics produces a lot of low confidence information. Then why do we keep on generating the low confidence information? So you can think of it, why it is so, is that the DNA binding proteins and the protein binding DNA, on the other hand, that is, these are studied in the computational sense from the statistical perspective, machine learning and biophysical perspective. And what happens in this, don't have that slide. I didn't remember that slide is not shown. So what I was just speaking about. So what happens is that some of the information generated by experiments is really low throughput, a small amount of information which is highly accurate. These are the experiments when you do a specific mutagenesis, and you get to know if you mutate this particular residue, what will happen. So these are the kind of high confidence information that you can collect from in vitro experiments or in the more recent CRISPR-Cas experiments. On the other hand, there are experiments where you do a large scale information is generated, such as the next generation sequencing, chip sequencing, for example. So, or ATIC sequencing. So this kind of experiments produce a lot of data, but all of this lot of data is not as accurate as you would be, for instance, the crystallographic experiment, which will produce an exact structure of a protein or a nucleosome or whatever it is. So on the one hand, you have information which is high throughput, but low confidence, specifically if you go to individual pieces of information there. And on the other hand, you have information which is of a low confidence, high confidence, but low throughput. I don't know if in what order I said, but I communicate the point that as the amount of data generated in experiment increases, the amount of confidence on individual piece of information contained in that also drops. So that is where the bioinformatics tries to help. It tries to connect the low throughput, high confidence information to the high throughput, low confidence information. So how we take the experimental data from mutations, how we take the experimental data from diseases, individual patients and individual cancers, and relate it to the gene expression profiles of thousands of people, or gene expression profiles under hundreds of conditions, or high-C experiments, where high-C experiment is done, for instance, if you look at the DNA interactions and the genomic in the high-C experiment, 3-dimensional interactions, the resolution of these information is 1000, at least 1000 base poor. You cannot see beyond 1000 bases there. On the other hand, in the crystallography, you can see individual atom. So can bioinformatics connect the two? That is one important thing. And the techniques that we can use in the bioinformatics are some of these techniques which come from physics, mathematics, and machine learning. So from the perspective of how I have contributed to the field of protein DNA interactions, I can say that there have been 3 generations of people, or 3 generations of the progress in the data set where this data has been generated. One is that earlier in the past, low throughput sequencing was being done, and crystal structures were being generated. Calorimetry was being carried out in the laboratories, biochemistry labs. And the second generation started with studying the gene expression, transcriptomes, microarray, and then later on other techniques of this collecting the gene expression data. And more recently now, you have high throughput sequencing, you have chromatin conformation capture. So there you can think of that, what kind of problems people were addressing, what kind of techniques people were using to address the questions of the time could be roughly divided into 3 generations. And as you see these 3 hands, they show the hands with the wrinkles and the hands with the promise. So let us say what we were doing in the first generation of these experiments. In the first generation of these experiments, we used to have a small number of transcription factor binding sites, analyze thermodynamics of mutants and protein DNA complexes. And that is where we started working. And first of all, we were just trying to identify what is a DNA binding protein. Because when people started getting to the genome and when people were already working on DNA binding proteins, they did not know all the proteins that interact with the DNA. They did not know all the transcription factors that will regulate a gene. So first interest would be to first identify the list of DNA binding proteins. So what is different in a DNA binding protein different compared to others? One would expect that DNA is a negatively charged entity. So the DNA binding proteins should have a positive charge. So we started looking at it a long time ago. And then we found that, yes, indeed, if I just look at the 3 properties of a DNA binding protein, or any protein for that matter, and if I calculate the total charge, if I also calculate the dipole moment, and if I calculate the quadrupole moment of these three properties, I will be able to predict large number of proteins or annotate large number of proteins correctly as DNA binding or not. But this requires a structure. So one advantage of this was that the dipole moment that you measure here is not really the true dipole moment. Even if you have a low resolution structure and even if the atomic positions are not well recognized, you can find a rough estimate of dipole moment. Why dipole moment is important is that although the DNA with the negative charge will attract the positively charged residues but many times we have found in the protein DNA complexes that a protein with the negative interface also interacts with DNA. Why this negative interface, negative surface of a protein or the net charge of the protein is negative, but there is still a protein DNA complex. So this is achieved by way of the polarization of charges. Some charges go on one side, the negative charges go on the other side and positive on the other side. Then the polarized side, the positive side of the dipole and formed in this way interacts with the DNA. So this kind of properties, a very simple model was able to identify as many as 90% DNA binding proteins in the database that were present at that time. Now the question is, if we do not know the structure, what kind of proteins bind to DNA? So we started looking at the sequence properties, and then in the sequence properties, the first property that comes out is the amino acid composition. And now, because to calculate the total charge on a protein, you really do not need to know the structure. You can calculate the charge by just counting the number of positive residues and counting the negatively charged residues. But what we found interestingly is that the DNA binding proteins are not really so very well enriched in the positively charged residues. So as this slide shows, in the left, that's if you just count the number of arginine residues, which are typically the residues in the interface with the DNA for proteins, we can only predict about 63% correct DNA binding protein. So the AUC of such a prediction model is just 60%. But if we count on the other hand, serine, we were able to find 70% accuracy for just predicting the DNA binding proteins. One would wonder why this paradoxical situation when DNA binding takes place through the positive charge, primarily with the arginine, what is serine doing here? So this is an enigma that we often face in biology, that we are actually looking for the mechanistic basis of a phenomena, but actually there is something indirect that is taking place there, which we often miss. So most of the DNA binding proteins have a signaling component, a part which carries them from one part to the other. So this part is not directly interacting with the DNA, but this part has an information that this is a protein which has been marked to bind to DNA, and it will be called. So in order to recognize such DNA binding proteins, you do not have to count the actual binding residues. Binding residues are actually few, but just the presence of a few binding residues, just the presence of a few DNA binding and the positively charged residues in a protein will not make it a DNA binding protein. There has to be some requirement of some other properties that will be able to take these proteins to the destination, the DNA destination. So if we then, of course, if we combine all the amino acid compositions, then we will be able to get a good accuracy and up to 82% accuracy from these models. Then next question comes that, okay, I know this is a protein that binds DNA, but which part of this protein binds DNA? This becomes important because if you want to design a drug, you want to know exactly which residue to target. Thus, protein targeting is not enough, because if you want to design a ligand, this ligand will bind to a small place in the protein. So there's an interest, can we predict the DNA binding residues from sequence alone? So this is the work that Michael presented in the context of protein-protein interactions as well. So we found this method. So we developed long ago these methods of predicting with the help of a neural network, the outputs, these 3 servers that came up from 2003 onwards, 2004 onwards. And we were able to predict not only DNA binding proteins, RNA binding proteins, DNA binding sites, RNA binding sites, carbohydrate binding sites, different types of sites on a protein. If we scan a sequence, and if we know what are the sequence windows at a given position, then the collective information of these residues and their surrounding residues together determines whether this residue will bind to one ligand or the other, whether this will bind to a particular type of dinucleotide, whether it will bind in the major groove or in the minor groove. All this kind of information could be predicted, and we developed these methods. So now the question is, why do we have to need to have a machine learning or a complex model for doing this? Because the role of the surrounding residues to a given residue, we want to know whether this residue is binding to DNA or not. This is an arginine. All arginine are not binding. Maybe arginine, which is surrounded by tryptophan, will help it to bind, but maybe arginine surrounded by serine will not, probably. So how will we know this? If we try to start creating these rules that are an arginine surrounding by this residue, and if the second residue is this, if the third residue is this, then the binding will take place. If we do this kind of process, then we will have so many rules to make and so few data that we will always find very good rules for the data that we are working on, but it will not work in any situation. So we need more complex models. We need models that can take highly non-additive behavior of the constituent properties or constituent features, which combine in a way that produce low amount of complexity and at the same time can model the non-additivity. So instead of having all the rules of combinations, that is 20 to, if I have an n residue window, I will have 20 to the power n features. So can I reduce these 20 to the power n features into a smaller, can I keep them in a smaller model? So neural networks are actually powerful because they use a hidden layer. They can introduce complexity of your desired degree. You can have 2 hidden nodes in the hidden layer. That will be low complexity. If you think your model requires more complexity, you can have 100 nodes. So you can easily manipulate the complexity of a computational model if you are using neural network, and that's why these models become so powerful. So on all these models, we use neural networks, and they proved to be very, very successful, and a lot of people liked them. That is why, it was mentioned, these are well-cited papers. So then secondly, after looking at the sequences, we started looking at what are the clusters of conserved residues, because conservation has always been often been implicated for any important property. Anything which is important must be conserved, and therefore conservation has been used as a measure for predicting DNA binding sites, RNA binding sites, all kinds of functional behaviors. The functional residues are apparently conserved, but these conserved residues occur as clusters, and this is what we studied in all the DNA binding proteins that were present, protein DNA complexes that were present in the PDB at the time. We published these works in 2008 and 2010. 2010, we looked at protein-protein complexes, in 2008, protein DNA complexes, and we found that the conserved residues occur as clusters. And now these conserved residues, when these conserved residues occur as large clusters, they act cooperatively. That is, if you remove one residue from this cluster, it is as good as removing all the residues from there. There are some conserved residues which sit separately, isolated. If you do anything to these residues, nothing happens to the complex. The protein DNA complex remains intact. So here we distinguish between the conserved residues which will come as clusters and conserved residues which come as isolated, and how they have a different pattern of contributing to the stability. And there are 3 types, different types of structures have different patterns of the clusters, and we investigated them. And we found there Okay, so this is not more in more details over there. We did find that we were able to find this proof that the conserved clusters, if we can identify conserved clusters, we will be able to most likely identify the correct interface that goes to the DNA. Second, this issue that was just being discussed before I started, the conformational change. So we looked at all the DNA binding proteins. We compiled a list of the bound and unbound protein DNA complex, protein DNA complexes in the DNA complex, and those the same proteins whose structures have already also been solved in isolation with DNA, that is the protein alone. And then compared and superimposed these structures. So we identified all the DNA binding protein conformational changes can be grouped into 6 categories, which are listed here. And these, for instance, if it is a rigid structure, you will find that the unbound protein and the DNA bound protein have exactly, they have no RMSD, or they superimpose perfectly well. If there is a little loop change, there will be loop movement, for example, here. So this is called local loop movement. When the protein comes close to the DNA, the arm, which has a positive charge, will bend towards the DNA. And if it has a negative charge, it will go away from there. So these play an important role. If this is important because if you are designing again a drug on a free protein, this drug will not work on when you actually take it to the environment where DNA is present. So we have to estimate what will be the conformational change likely to take place. If we do not have a complex structure, we have to estimate what are the likely flexible regions, changing regions, and where we have to focus on the potential deviations from what we are currently doing. So that's why you have docking for complex, the rigid docking and the flexible docking. So these are the other conformational changes, like secondary structure might change when the protein comes in contact with DNA. There may be order to disorder transition or vice versa in them. There may be a complete arm, helical arm, which goes from one side to the other. And there may be a more complex, the quaternary level conformational change, where two monomers or three more monomers come together in a particular conformation. They expand, they open, close, all kinds of phenomena take place. Especially this happens in the case of the immune-related or pathogenic-related the molecules. Then you have interdomain or quaternary structure changes. These are the examples: restriction endonuclease, not I, and the Y family DNA polymerase 4. So these are some of the examples. In terms of the implications to the stability of the protein DNA complex, we found that if the RMSD for the open close conformation is higher, 7.9 angstrom change happens in the RMSD when this type of conformation changes, compared to the secondary structure change, where it leads to just about 1.5 1 angstrom change in the RMSD. And the large conformational changes occur in only the two groups, then interdomain and the open close conformations. In this slide, I show here that if you look at the specificity of a protein DNA complex, that is a protein which binds to a specific DNA sequence, and there are those proteins which will bind to any DNA sequence. So this specificity is directly related to the conformational change. Greater the conformational change, more specific is the complex. So it is programmed into the proteins that they have been left alone in a flexible state, and they will bind to a specific conformation only. So the conformational changes, particularly OC and ID, the conformational changes occur in highly specific complexes. In the non-specific complexes, these phenomena do not occur mostly. And when we look at it in a greater detail, individual PDB, we find that this high conformational change of 6 angstrom was observed in this case in 1 BAM, which and compared to this, there is a relatively small conformational change here. And if you look at this y-axis here, this shows the complex stability. So the complex stability is high for this complex, which underwent 6 angstrom conformational change. So that means the change between the free and bound DNA bound complex is being utilized as a kind of switch or as a kind of key to make the complex stable. So the protein is moving around freely, and when it comes close to the DNA, it changes the conformation and makes a very strong complex. On the other hand, the proteins which are rigid, they come in contact with DNA, they do not form very, very strong complex. So they are strong in themselves. So perhaps that is why they do not form very strong complexes with the DNA. So this is what happens. And now if we look at the nature, the reversal of this problem, we were looking at so far what kind of proteins bind to DNA, what kind of regions in the protein bind to DNA. If we reverse the problem, we are now looking at what kind of DNA actually attracts proteins. In the genome, every region is not a protein binding region. So what are the properties of the regions where a protein binds, if it is a genomic phenomena? It may not be genomic DNA, it may be a floating DNA, it may be cytoplasmic DNA. There can be other types of DNA, but the genomic DNA is where we are more interested in. So if there is a genomic DNA and if there is a transcription factor, what are the locations in which it will bind? So what are the DNA properties which determine whether there will be a binding? So some review where we look at the base, those who do not know the DNA structure, there are three broad structures of B, A, and Z DNAs. And then they have different kind of stacking patterns, and that allow different types of protein structures to interact with them. And compared to this ideal bDNA structure, the actual folding of the DNA in the genome may undergo some shear, some buckle, some stretch, propeller, stagger that have been defined as if these are, in this case, two base pairs. If the base pairs are not completely aligned, there may be a shear. If they're bent slightly in their planes, they're called buckle. So these are the conformational properties that have been defined. And then in a real DNA that is coming from the three-dimensional structure or the crystal structure, we can measure that in a given position, what is the shear, what is the shift, what is tilt, etc., for the base pair and the base step. So these properties are sequence specific. All bases do not undergo the same level of these changes. For example, if you compare the A DNA and B DNA, you will see that the buckle in the A DNA is minus 0.1. On the other hand, the B DNA has a 0.5 buckle. So all these parameters, so they are called conformational parameters of a real DNA. These conformational parameters of a real DNA are sequence dependent. They are different in A DNA, B DNA, and Z DNA, and they are also sequence dependent, as it is shown here. Because they are sequence dependent, the different sequence patterns, when two sequences stack over the other in a double helix, they are differently stable. They're not all equally stable. One level of stability difference comes because G can form a triple bond with C. So GC regions are more rigid, AT regions are less rigid. But not just that, even the stacking patterns, if GC is sitting on GC and GC is sitting on CG, then they form different kinds of energies. For example, if you see there on the top, you see CG sitting on GC, this will give rise to minus 61.0 kilocalories. So this is a highly stable stacking. On the other hand, if you look at this GC sitting on CG, where this is minus 40, although both of them are G and C, and the triple bond is likely taking place here. And if you go further down, so GC sitting on identical, that means if the base pairs are GC are separated on the two sides, G on one strand and C on the other strand in successive positions, then the energy is even less. So these stacking energies go beyond the double bond and triple bond energies. And this is realized by way of the conformational changes, because some of the sequences are flexible enough, they will adapt certain buckle, they will adapt certain role. And these are some conformational subtle changes will take place in these DNA sequences. When you put one sequence, it will have one change in the conformation. If you put another sequence, there will be another conformation change in the conformation. This change in conformation or these stacking energies based on the sequence is very important. Why it is important is when the transcription factor is scanning the genome, it is actually not seeing the base, because the DNA, double-stranded DNA does not have base on the surface. It has only the backbone on the surface, sugar, phosphate. So the transcription factor is not seeing the base. It does not know that this sequence is Tata box. How does this then Tata box binding transcription factor find its Tata box? It will find through what you call indirect energy, indirect readout, or by the way of the shape readout. That is, that there is a base sequence inside, and this base sequence will slightly alter the double helix. And these alterations in the double helix can be recognized by the sliding transcription factors. So this is a beautiful arrangement by the nature without having to expose the nucleic acid bases to these transcription factors. It allows transcription factors to recognize what is the sequence hidden there. So recognizing these things, and we are quantifying the interactions between the protein DNA complexes. We have worked on collecting all the protein DNA complexes and tried to see what are the nature of conformational changes which favor to provide specificity to a given complex. So this web server that came a long time ago again. So this URL is also not correct. Please don't copy. Those of you who are looking at it, I'll give you a separate URL if you need. So this allows us to tell if I have a large number of sequences and I have a protein DNA complex, and I want to know from these DNA sequences, which of these sequences can form a similar complex, or which of these sequences can form a more stable complex than the protein DNA complex that I have. So for that purpose, we can use this server. We will try to compare the conformational preferences that each of these sequences has And from these conformational preferences, we can infer whatever is more suitable for the current geometry or topology of protein DNA complex will be the most preferred sequences. So this allows us to identify novel targets, novel DNA sequences, because if you just compare at the sequence level, you might find two sequences are similar or two sequences are very different. But if you convert them to the conformational parameters, they might show different patterns of similarity and difference. So that is what has to be captured, and we utilize this in actual collaboration through experiment with experimentalists, and one of a non-canonical binding sites of the SAA gene was discovered by this procedure. So the conclusion from this part is now that both protein DNA have unique sequence and structural features, and they enable them to recognize their partner molecules, amino acids, type, evolutionary profiles, conformational states, conformational changes are crucial for the DNA binding proteins and confer the function and specificity. In intrinsic conformational dynamics that I showed you in the readout web server is critical for this process. So now second generation, I now to move to how the gene expression patterns, what kind of gene expression patterns are observed in the case of DNA binding proteins. So here we have an analysis. We found that if first we looked at the global data, we collected all the samples of gene expression that are there in the gene expression omnibus and looked at the genes that code for DNA binding proteins and looked at the genes which do not code for DNA binding proteins. And we found that yes, the expression level of the DNA binding proteins is higher compared to the non-DNA binding proteins, and the p-value is excellent. So we are impressed. But we should never be impressed by such an artifact because this is an artifact. The artifact is because if you take any functional annotation, the genes which have been functionally annotated are likely to be expressed at a higher level compared to those which have not been annotated. Because these poor genes have not been annotated because they were not expressing well enough. So any annotation you take you will find that the highly annotated sequences are highly expressed, and poorly annotated sequence or poorly understood sequences are not so poorly expressed because people have not paid so much attention to them because there was not much information available about them. So this is the case. So how do we know whether this observation is really useful or this observation is an artifact of the data analysis? So we looked at the comparison around the co-expression data also. And so I'll show you that. No, I don't have that slide. So we found that there are many genes which are whose average expression level is not high, but they are DNA binding protein. So one way when we have a complex issue to deal with, when we have too many features to talk about, one way whether these features are relevant for my function or not is to take those features and try to predict what percentage of proteins I can predict from there as DNA binding. So this plot shows that if I take the gene expression signature in the global context as the input for a protein, whether this protein or this gene will be a DNA binding protein coding gene, then I can get a relatively modest prediction accuracy of 64.5%. But when I take sequence properties, I get an accuracy of 84% in the best case scenario. So that means, does this mean that gene expression profiles have no value in predicting DNA binding proteins? No, because gene expression profiles are actually context specific. Sequence tells us, okay, this protein has the ability to bind to DNA, but it will not tell us when will it bind to DNA. If this protein is not available inside the cell, what is the use of telling that this is the DNA binding protein? As we say in poetry, said, "Apna wahi hai waqt pe jo kaam aa gaya." If this protein is not there, if its partners are not expressing there, if partners are not present, then even if this protein is highly DNA binding, it has a strong signal of DNA binding, it is useless. So but how do we get this information? So that is where we distinguish between the potentiality of a biological function and actuality of the biological function. The potentiality of a biological function is encoded in the sequence and structure, whereas the actuality of the biological interaction is encoded in the transcription environment of that particular protein. So if the right kind of genes are also simultaneously expressed, it will do a function. And that is how the whole biology proceeds, where even from the start of the development, when one organ is formed, another organ is formed, the genome is not changing. All these proteins are encoded. All the transcription factors, what is called the pluripotent genes and the differentiation genes, etc., all stemness genes, all these genes are present there, but they are not expressed, or they are a right kind of set of network is not expressed. So this is where we have to distinguish. What we do not have to just know the annotation of a protein, we also have to know the expression conditions or the cellular conditions in which that sequence is present. So we found that we could annotate some of the DNA binding proteins from gene expression, which could not be annotated by the sequence. So there were some proteins which had a weak signal to be DNA binding proteins, and our method was failing to capture them as DNA binding. But when we add to them the gene expression signature, we found that they are supported by cofactors. So they are weak binders. Their signals at the sequence level are weak, but if they have the right kind of expression environment, they will perform the DNA binding function. So the second conclusion from this analysis comes that the gene expression patterns and co-expressed genes may alter the behavior of certain proteins to bind DNA in this case, but in any function in general, when the sequence itself does not contain a strong signal and a strong enough signal. So biological function of DBP is not encoded in sequence and structure alone. It is also encoded in the gene expression environment and the environment of the entire cell, and the maybe the metabolites, etc., also, but we have not looked at them. So now the final part of my presentation today, the age of the high throughput and big data. So what are we doing in the big data? So we started with this study. And when we started looking at ChIP-seq data, the first study that we did was this, that we tried to look at the STAT3 binding sites from ChIP-seq experiments. And 4 different experiments were producing different results. And we tried to see whether the sequences which are bound in one cell, are they similar to the sequences bound in another cell? So in 4 different cellular conditions, you find that only 35 of these thousands of sequences were binding to the STAT3 transcription factor under all 4 cell conditions. Large number of them, 1,547, were only binding in the macrophage environment. And 2,148 sequences were found binding only in ES cell environment. So how is it that we say that this is a protein, this is a DNA, this is how it interacts. The same DNA is present there, same protein is present there. Why it is not binding there? Why these sites are different? So we have to look at what are the environment in there acting. So we try to look at this from the perspective of the, I showed you earlier that there's a readout mechanism where the DNA structure plays a role. So we cannot simulate the entire genome to look at the role of the DNA shape. So what we did was we took a unique set of all tetramers, and we performed the molecular dynamics simulation for all of them, all the exhaustive sets. And then for every pentamer that occurs in these exhaustive list of sequences, which was done in a 12-mer environment, so we could get pentamers there. So for every pentamer in our database, we try to create a conformational ensemble. Okay, 20% of the times, this sequence will have a shift value or a twist value here. And 20% of the time, this twist value will be slightly higher, etc., etc. So defining this, so we defined what we call the conformational ensemble of the genome. And the entire genome in this way can be simulated. So we can tell about the dynamic environment, the conformational dynamic environment or shape dynamic environment of the entire genome in a real time, which is prohibitively large to deal with to simulate in reality. So here it shows that if we do this kind of a shape-based analysis and sequence preferences of the conformations for each sequences, you find here that the sequence similarity does not correspond to the shape similarity. So for example, you have on the top in this slide, TAA and TAG. There's just one base difference here. So they are in the same cluster. But you will find here, if you look at the 1, 2, 3, 4, 5, 6, 7, 8th TAT sequence, there is also just one base difference. So the sequence here, TAA, and this sequence, TAT, they have just one base difference. But still they come in a different cluster. So that means if we just look at the sequence level differences in these nucleotide sequences, we will not be able to capture the right kind of patterns. We will not be able to group the binding site versus non-binding sites. We have to go from the sequence information to the shape information or the structure information. And then when we use this property in the motif flanking regions for the transcription factor start site in STAT3, we found this result. So using our tool, what we call our tool is DynaSeq. In this tool, we were able to predict a large number of a very good degree. This shows just a 64% AUC, but this is quite good because I showed you in that slide, there are just a very small number of sites, 35 sites, which are common to all cell types. So many sites which could be predicted to be in the correct cell type. And now if we look at the 4 different cell types, we also observe that the bound regions around the motif have a different pattern in different cellular conditions. And you can think of that the actual motif is located in one place where transcription factor will bind, and this will be surrounded by modular regions. And these modular regions are about 50 base size, and each of these modular regions follows a different computational model, in this case, a linear regression model. So, and every different cell type has a different shape. So looking at this shape, we can also characterize different cells into groups, and we can also tell what cells are similar to each other, and we can estimate the behavior of the transcription factor binding sites from the non-motif regions, because non-motif regions are generally considered to be not informative at the sequence level, but we found that at the structure level and the conformational dynamics level, they're highly informative. So when we look at this in further detail, there are transcription factors which serve as pioneers. They go and open the chromatin. Then there are settlers and migrants which bind to the open chromatin. And we looked at this behavior of this in terms of these conformational preferences of the sequences that are contained in them. And we found that it looks like that being a pioneer is not the property of the pioneer itself. Protein property is the property of the target sites that it binds to. So if the pioneer is likely to bind TATA or pioneer is likely to bind TGAT, it will have that kind of a property. So the information is actually stored in the complementarity of the sequence, not in the entire structure of the pioneer. That is what we observed from this data. I'm not explaining this data in detail, but that is the supportive data that comes. So more recently, because I started saying in the beginning that we have lost our address of molecular interactions is perhaps lost. I showed you some of the results that reminded us that these actually we have not stopped working on molecular interactions. This is an important area of the research today. So one of these is most recent collaboration that is going on with the Manchester University, where people are trying to understand how the HOX proteins, there are 2 transcription factors, HOXA2 and HOXA3, they bind their targets and they actually take place, they determine the organogenesis in the mouse development. So in the process of development, some of the transcription factors act early, some act later, but nobody knows what is the difference in the binding sites of the transcription factors. And we applied our tools, DynaSeq, and did additional analysis. We found that there are some motif-free positions in the HOX targets, and there are some motif-containing positions in HOX targets, and we could further coarse grain our ensemble to, instead of 5 bins to 3 bins, and using them, we were able to distinguish what are the conformational properties that determine the specificity of HOXA2 versus HOXA3. This is a very exciting result that we are currently working on, and I hope this will see the light of the day of publication very soon. Finally, the conclusion from this is that the conformational dynamics of target binding sites and flanking regions are crucial for TF recognition. Cellular specificity may be significantly impacted by the long-range 3-dimensional organization of genomic DNA. And as I showed you in the 3-dimensional property structure of the DNA, genomic DNA, and the sequential cooperativity of TFs may be encoded in their target DNA structure and not in the protein itself. So this is the final outcome from the 3rd generation that I just mentioned with you. I end here. I do not have a thanks slide. And of course, all this work has been done by many other people, and I've had the luxury of showing them here. I acknowledge them anonymously. Thank you.
Thank you so much.